another episode of Bioconversations Demystifying Science. In our journey so far, we have explored the fascinating world of process sciences and how biologic products come to being. Today, we pivot slightly to focus on an equally vital piece of value chain, the analytical sciences. When it comes to making biotherapeutics, it is often said your process is as good as your analytics. That's because analytics play a crucial role in confirming whether the biotherapeutic that you're making is actually the correct one, it is safe, effective, and consistent each time you make it. In this episode, we will explore how analytical scientists assess the identity, structure, and quality of these complex biologic drugs through what is known as physicochemical characterization, a word that's mouthful, but we will demystify it in this segment. Joining me today is Dr. Navaratna Vajpai, who earned his PhD in structural biology and biophysics from the University of Basel, Switzerland. He joined Biocon Biologics a little over 8 years ago and is currently the project group lead in Analytical Sciences function at Biocon Biologics, Bangalore, India. Welcome to Bioconversations, Dr. Navaratna.
Thank you, Dr. Kavita. It's an absolute pleasure to be here, and I'm glad that you have invited me and happy to give my insights on analytical sciences function.
So before we segue into the actual topic of discussion, few icebreaker questions for you so we get to know you better.
Sure.
So for example, you have had a rich background in biophysical analysis and you moved from Europe to India. So what was the motivation to actually pursue a PhD in a subject that is so unique? What drew you to this field?
Thank you, Dr. Kavita. So my interest for structural biology actually started when I was pursuing my master's in chemistry, but I took a subject which was very relevant not only for chemists but also for biologists.
Mm-hmm.
And it is then when I understood the importance of knowing and understanding the physics of folding of proteins. It has definitely helped me first to learn about proteins, but also about their interactions with other molecules. So my training actually formal training began in Switzerland during my PhD. And I got a chance to work in collaboration with Novartis Pharmaceuticals. And that has developed or enhanced my understanding about structural biology of pharmaceutical proteins.
So you have worked very closely in academia as well as industry and guiding PhD students towards their thesis while working in industry. So what are the key differences that you may have noticed and how these understanding of academic network helped you professionally and vice versa?
Very relevant question, especially when we believe that a joint collaboration between academics and industry only can synergize the development of more complex drugs which are actually coming into the market as well, also to address patient needs.
Mm-hmm.
So the academics gives you a flexibility to go around and experiment more, discover new things, which sometimes in an industrial setup may be challenging because there is a different need and requirement of an industry to address patient needs and also to commercialize products, to move things into the market, help reaching patients. So I think we need to leverage information from both strengths of both and look for synergistic effects out of that. Academics definitely gives you an opportunity to find best of the talents who can think out of the box many times, and industry uses that information to develop new modalities towards addressing unmet needs.
Now that we are on this topic of physicochemical sciences, or characterization that we are going to discuss in this segment, it's a very complex and a mouthful, right? So can you help us understand whether physicochemical is essentially physics and chemistry blending together to understand the molecule, or what exactly do you do in physicochemical analysis?
Very rightly said. Physicochemical means we need to understand both physics and chemistry of molecules. And in this case, these are molecules of our interest, which are protein molecules, highly complex in nature. And because they are very complex in nature, what you would need is also multiple lenses to look at these molecules from all angles. And one of the reasons these are not only complex from their structure point of view, but also the way they are produced or generated.
What is the size of molecules, the range of sizes that you study in your physicochemical analysis there?
So we work on many different sizes of molecules. At Biocon, we work on insulins or even small GLP analogs. At least the biologics division had worked on GLP analogs, which is like 4 kilodalton, and you move up to 150 kilodalton to 200 kilodalton proteins. So these are very complex in nature, and you would need multiple orthogonal tools to see from all angles.
So orthogonal means battery of tests, you mean?
Orthogonal means you look at the molecule from different angles and confirm the information from one and leverage that information to confirm the overall structure and function of the molecule.
So when you say information, what are the things that you study in your analysis?
So when we say physicochemical analysis, the first part is to do separation of these products, which is the main product of interest,
Product of
biotherapeutic, from its variants, its impurities. So you use a lot of separation technologies like HPLCs.
So what is HPLC, if you can define it?
Yeah, HPLC is like a very fine filter. And if suppose I give you multiple molecules of different sizes and you want to segregate them, then based on one interactions or the others, you can actually separate these molecules individually, and each one of them will be a representative of that kind of molecule.
Okay. And what are the other? So basically, you have a toolkit.
Yes, correct. We have got a toolkit, and the other toolkit involves, say, mass spectrometry.
Now that is another complex word. What is mass spectroscopy?
Oh, wonderful. So mass spectrometry.
Actually, what is spectroscopy?
Right. So spectroscopy provides information of molecule based on certain phenomenon called resonance. So in that, what you do, you excite a molecule. It goes into its excited state, and then when it comes back, it emits some radiations. Those signals are identity of that molecule, and different ways you can do that. One of those ways is NMR and FTIR, which are very commonly used.
When you talk about signals, so each molecule emanates a different signal.
Yes.
Which
helps in identifying and understanding of that complex molecule. So you look for those peculiar signals and you connect that information, and that is why I said orthogonal. So you do multiple different such tests to actually build on that information to obtain.
So now when it comes to mass spectroscopy, what do you do in mass?
So in mass spectrometry, what we do, especially in cases of proteins, we actually take larger molecule, we measure it, and mass spectrometers today are extremely advanced tools. They can provide very accurate mass of these molecules. Now these masses are also an identity of that molecule. They are absolutely accurate. So you get once you get that information, that is the first step to say you are in the right direction. Now, when you want to further go deep into the molecule, you digest that molecule. Digestion means you cleave that molecule into smaller parts, and each of those peptides, which are actually the building, or amino acids, which is the building block of these large proteins, you actually look at their signals, and then just like a jigsaw puzzle, you build on those signals, connect those information, and build the amino acids. So it's like a fantastic way of solving a jigsaw puzzle. And the scientists in my team, they actually enjoy doing it, and sometimes they provide very relevant information.
That's very nice. So you have these techniques, and then you mentioned something like CD spectroscopy, FTIR, or
Yes.
NMR. In brief, what would they be?
So they all provide you a very relevant information. So it is like if you look at an elephant and say suppose 5 or 6 blind men, they are being asked to look at an elephant. They will all tell information what they can touch and feel. Now,
That's a good
these different tools actually provide us a very, very similar information. So they will just connect information and and then demonstrate that this is an elephant.
So that's very good that you have a battery of tests that you run on your molecule to understand its structure, the way it folds, and give the complete profile of the protein. But if I may know, why is it necessary to do all this? And what is the information that you're trying to look for when you're doing this?
So, very relevant question. See, these biopharmaceutical drugs, which are mainly proteins in this case, they are produced through a very complex process. And this process needs humongous number of controls during the entire, from the start to the end of the development.
So tightly monitoring how you're...
Very tightly monitoring. It requires a high monitoring of the process. So when we have to do that, we actually identify critical attributes.
Attributes that are critical in what way?
So these are critical because they will help defining the safety and efficacy of your molecule.
Okay.
So what you do, you ensure that the product of interest is the only one which is pure, very pure. The product of interest is in the right conformation. The product of interest has a right folding. And therefore, when you obtain that information at the end of the entire development, this is the information you pass it on to your manufacturing and quality colleagues.
So why is it important for a protein to fold properly? What happens when there is misfolding?
The structure, and this is where my learning of structure biology helps. The structure governs the function. So if your protein, for example, has not come into the right shape, then in the right orientation, then it can lead to a different kind of immune reactions inside the body. For example, unfolded proteins are known to create neurodegenerative diseases. Mm-hmm. So you have to ensure when I am delivering something to the body, your product goes in the right shape so that it can do the function it is intended to do.
So hold your thoughts here. So the next part of our series would focus on the function of this molecule once it goes inside the body and what it is going to do. So we will not discuss the in vivo attributes of a misfolded protein because that will be in our next segment. So you can continue.
Right. So suppose, the interesting part what you asked about is why they are so important, these attributes to identify. So these attributes, as I said, we need to monitor them across the entire development, but also during the manufacturing cycle. So
it doesn't end once the molecule leaves the lab, but it is a continuous
process.
Behavior. Yeah.
It is a continuous process. And during the entire course of this development, we also ensure that the product is also stable across a long period of time. Because we need, once the manufacturing is done, product is produced at one site, it is also to be transferred at multiple sites. Mm-hmm. So during this course of time, we also do a lot of additional stability studies. And these stability are also done in a way that we induce from outside some external...
You give an external stress.
So that we can mimic or simulate situations which are real and probably also our product would see otherwise. So we stress our product through heat, we stress our product through, say, chemical reagents, we stress our product through light, we also stress our product through agitation.
Right,
and all these different tests, under all these conditions, we also test, is my product degrading faster or so?
Yeah.
And we identify root causes, and then those help us in actually defining the guidelines how I can keep my product more
Give
intact.
recommendation on storage. So storage becomes extremely critical.
Storage becomes extremely important.
Even at the patient's end.
Even at the patient's end, and for biopharmaceutical, it becomes even more strong.
Correct, because temperature has so much of an impact on the way your molecule
Proteins
behaves.
are delicate. You have to understand proteins are very, very delicate molecules, and they are very dynamic in nature. So little here and there can also stress them. So therefore, stability conditions are extremely important.
So that's why it's even more important for a patient to go through the leaflet that comes on storage.
Oh, absolutely. I think for everyone who is doing this, whether it's patient, whether it's the practitioner or the people who give pharmacies, they all have to take care of this important part of storage.
So now this is one part of analysis. Now the patient receives the product in a particular presentation, like a vial or a cartridge. So do you also study compatibility of the protein or your therapeutic agent with the containers?
Oh, yes, absolutely. This is also very, very important study, because it is quite possible that the rubber cap can interact with your protein of interest, or the excipients during agitation is subjected to these kind of interactions, which may lead to, say, rubber particles coming out of your caps, or anything else, maybe glass particles also coming out of your vial, and which can be extremely dangerous. So for those things, what we do simulation studies. We also study in not only on a very long term, which is the stability we define, but we also do stress conditions again under to establish that no such rubber particles come out of those.
Its way into the product.
It's a swing product, and these are very, very important studies to conduct, and we do it all the time.
So now that you have the presentation, you have your product in your container, and before it goes out into the market, what are the additional tests do you do? Are there any tests that you do to ensure safety and quality of your product?
Yes. So once the product is manufactured, so as I said in my early discussion, that there are steps to control and monitor the process. While that is done, the final product or the finished product, after it is filled in the presentation, is again tested for multiple attributes. These attributes are generally such as you check the pH of your product, because it defines the stability. You check your subvisible particles or visible particles.
So what are these subvisible and visible particles?
So these are submicron particles or micron particles, and you can't see them with the eyes. The visible particles are ones which you can see, and sometimes you probably have seen the products are unsafe because you see some particles, and immediately the recommendation from the physician is not to use that. So we ensure that such subvisible particles or visible particles do not get generated during the stability period. So these are very, very important. And there are guidelines. There are ICH guidelines, which is guidelines which are followed across globe. And there are also pharmacopoeial guidelines, which are also followed across globe, which is what we also intend to follow. Every product behaves very differently. So you have to maintain or understand individual product and define guidelines, define product behavior accordingly.
So how long do these tests typically take before, after it's manufactured and before it is released? Typically.
So there are different tests which are conducted, and some tests which are very short. You can do it in hour, minutes to hours. But some tests take longer period of time, especially microbiological testing, sterility testing. They may take longer, sometimes 1 to 2 weeks for every time a batch to be released. And the compilation of the entire activity takes like 5 to 7 days. So, and then there is a quality check, because that's a very, very important work to do. It's for patient, and we ensure that the quality is correct. So every result being tested by the analyst, but then also evaluated and assessed by a reviewer, which is not only the reviewer from the same technical side, but also a quality assurance officer. So it is then evaluated, tested, and ensured that the quality is correct. Altogether, it takes between 3 to 4 weeks one batch takes 3 to 4. So drug discovery is not very easy. It's a complex thing.
Yeah, I understand that it's quite cumbersome with multiple peoples involved and multiple techniques involved, and then you have to piece all that information together to ensure that your molecule or your product which is landing in the patient's shelf is as safe and efficacious.
Absolutely. Very, very important, very critical for us. Yes.
So, thank you, Dr. Navratna, for sharing your insights on this particular topic. But before we wrap up, we would like to we would like you to indulge us with some rapid fire questions. So, you are a thorough scientist. You eat, breathe science for as much amount of time I've known you. So, how do you unwind after a busy day?
Oh, I sometimes I go out with my friends to enjoy evenings to for food. I am actually a foodie, so I enjoy food. I go.
Or you just eat?
I cook, but I also go outside, and I love all kind of food. I'm a vegetarian, but I find a lot of vegetarian good place nearby my home, so that's something which I enjoy. I also watch a lot of movies, so some of those things I help me to unwind myself.
Because you need to keep your creative
Yes,
spirit
yes.
alive, right?
And I actually have a lot of good friends, so I talk to them over phone. I call my family, speak with them, and it's fun, especially talking to young kids. It's so much fun to actually talk to them.
So one particular aspect about you is you have a great connect right from a 3-year-old to maybe an 80-year-old. How do you keep that alive across generations? What is the way in which you connect with people so effectively?
Something which I have learned that human connections are extremely important, and what I have always tried to do is to go to the levels when I meet people. And one of my brief experience I'll share. When I moved to Switzerland, I am I'm a fan of cricket and born in India, so you can imagine cricket is flows in our veins, and I used to play well as well. So when I moved to Europe for my PhD, everybody always spoke about football, and they would talk about clubs, they would talk about leagues, and I had absolutely no clue about. So for initial months, I actually found myself alone, and this is exactly what I learned, that no, if I have to stay here and do well, I also need to connect with people.
Correct.
So first thing I started doing is to start watching football matches. So every
So
morning,
activity is actually a good way to connect with people. When you move to a new country
I
or a new
started going
city, activity clubs.
to pubs to watch football games, and then that helped me to actually just start talking about their language, because language was different, food was different, and everything. I grew up in a very humble, modest family, so a lot of these foods were also very alien to me. But I think over the
You
period,
did well.
I developed the interest. I also started enjoying football, and a lot of games I started watching with them. So that helped me to connect people. So that's very, very important, and I do the same now.
And it comes very organically.
It comes organically, and I'm glad that I could build a lot of good friends over years, from small child to quite aged, and I enjoy talking to them.
Finally, one last question. If you weren't a scientist, what would you have been, or what career option would you have pursued?
So I studied in a military school.
Wow.
My father was also in Air Force, so this was one of the things I always wanted to do. I appeared for exams. I could not sail through, but uh, that's the way. But I learned something very important, that
In a military setting.
in a military setting, I learned that when the idea for me was always that I wanted to serve the nation, and when military thing did not happen, I thought that whatever best I can, I will do. So I went outside, learned things, and I
You
came
brought
back,
that knowledge home.
and I brought that knowledge home. Every time people ask me, would you stay there or come back, I always thought I have to come back and do something here, and I'm glad I did it. And I thoroughly enjoy what I do. So that is also the key for the long sustenance in the field.
True. That was amazing. I think we learned a lot more about you, more than just the science you do, but the person that you are and the grit and the passion that you have for your science. So it's been extremely amazing to have this session with you. Thank you so much.
Thank you so much.
And that's all from this segment of Demystifying Sciences. Until we meet in the next segment. Stay tuned.